Alrighty, chapter 21 is about blood vessels, and it is our third and final chapter in the cardiovascular system. Now, sometimes you would refer to the blood vessels themselves as just the circulatory system. That's absolutely appropriate, but they really are part of this larger cardiovascular system. They don't work on their own. They work in tight unison with the heart. We'll see that particularly when we get to the physiology aspects towards the end of the chapter. You'll notice maybe the overlaps with chapter 20 in the heart when they started talking about regulation of things like blood pressure. The two work together, and so we'll really hone in on that here in chapter 21. We are going to start off with some anatomy of these blood vessels. So some of this is fairly straightforward. Hopefully you already realize things like when blood leaves the heart, it moves into arteries, followed by arterioles. That might be a new one, right? Smaller arteries called arterioles. Eventually down here into capillaries, and then it's returned to the heart through things like venules and veins, and eventually back to the heart. Now, I will say the superstar of this show, the often overlooked capillary. And I say that because really, if we think about what the cardiovascular system is trying to do, it's trying to circulate blood so that things like nutrients and oxygen, hormones, all of these things are getting delivered to tissues. And that delivery takes place at the capillaries. That's where diffusion happens. And remember, diffusion happens over very short distances, really no more than maybe 1 or 2 cells away from a capillary can get the oxygen and nutrients it needs. And so that means you have to have capillaries that reach out to almost every single cell in your body. You have something like 10 billion capillaries, 25,000 miles of capillaries in your body. So we're definitely going to spend a little time looking at how those capillaries work. See, somebody has to get excited about capillaries. Okay, to get there, we have some vessels to look at. Now, overall, you should start to notice some patterns, differences between veins and arteries, right? Looking for that. And you should also be looking for, we're going to kind of hone in here on the 3 layers that we're going to find in blood vessels. We'll find these in arteries and veins. We'll find these in different size arteries and veins, right? But there are going to be variations. So let's start by looking through these 3 tunicas, these 3 layers that are going to be in our blood vessels. Now, one thing that's kind of fun to point out here is this structure called the vasa vasorum. As we start looking at blood vessels, it's a little counterintuitive, but you have to realize they're going to end up being so thick, right? I mean, this tunica media in some of our vessels might be 50 cells thick, too thick for diffusion. And so just like the heart had its own blood supply, so do blood vessels. And so this is referred to as the vasa vasorum. It's the blood vessels for the blood vessels, which is weird, right? Kind of wild. Okay. Otherwise, what we have on here, we're looking at our 3 different layers. So the tunica adventitia is going to be the outer layer of blood vessels. The tunica media is in the middle, and then the tunica intima is on the inside. It's connected. It's intimate with the blood. Let's start there. So the tunica intima, the very innermost layer is referred to as the endothelium. You might recall endothelium is really just a fancy way to say simple squamous epithelium in the circulatory system, right? So endothelium is another way to talk about epithelium here in the cardiovascular system. Just outside of that endothelium is a basement membrane. You might remember that from the skin, just a thin layer of connective tissue. Outside of that is a structure called the lamina propria, more connective tissue. And then finally, the internal elastic membrane. Elastic fibers, this is also connective tissue. So at its simplest, right, the tunica intima is really endothelium and some connective tissue that surrounds it. You'll see some differences here, whether you're in arteries or veins. Typically in arteries, this layer has more elastic fibers in it to help absorb the stress of that kind of bolus of blood leaving the heart. Okay, tunica media, right? Media, middle. This is going to be the muscular layer of our blood vessels. This is smooth muscle, right? Not skeletal muscle, not cardiac muscle, smooth muscle. This is going to be super important in our blood vessels being able to do vasoconstriction and vasodilation. We'll talk about that more. The tunica media also has some connective tissue, this time in the form of what's called the external elastic membrane. So internal elastic membrane on the tunica intima, external elastic membrane is in the tunica media. It's not the very external surface of our blood vessel, but it is outside the internal. Okay. The tunica adventitia, then, also sometimes called tunica externa, this is the outermost layer of our blood vessel. It's a connective tissue covering. It helps anchor blood vessels to adjacent tissues. So in a couple weeks, when you start dissecting blood vessels in the cat, you will start to see this. All the blood vessels are kind of tied in to the muscles and things they run near. And again, blunt probe, just gently separate. You don't need a scalpel. In arteries, this tunica adventitia will tend to have more elastic fibers, so there'll be a lot of collagen with some elastic fibers. In veins, you'll actually see that sometimes the tunica adventitia has a little bit of smooth muscle in it. Okay, I pulled this out of a previous text because I thought it was a pretty cool look at histologically, so this is an actual microscope view of an artery and a vein, and then an artist's rendition of what we're seeing there. So, and then down here at the bottom, they give you kind of a nice linear, tabular form of the same information if you think more that way. A couple things to point out here. Arteries tend to have thicker walls. A lot of this has to do with the tunica media, so layers of smooth muscle, and you can see that here in the picture, how much thicker that is. Arteries also have a lot of elastic fiber in them, and so they tend to recoil when they're not filled with blood. And so you'll see that this guy is kind of small and round. He's recoiled, where veins end up just kind of getting loose and floppy without blood. The other thing that recoil does is kind of throws that tunica intima into little folds, right? In real life, this would be a smooth vessel wall so the blood can flow along unimpeded, but without blood inside. It tends to recoil there. What else? Oh, you can't see it on this picture. Veins have valves. We'll talk about that more when we get into the veins specifically. All right, now, so if you have those three layers, tunica intima, media, and adventitia, or externa, either way, that's going to differ, not just between arteries and veins, but also within arteries and within veins. And so let's take a look at this fairly briefly. All right. So as we think about arteries, one of the key things here really is that consideration of things like vasoconstriction and vasodilation. We're going to be able to make huge changes in things like blood pressure based on what the arteries do. And so we'll see the sympathetic nervous system causing vasoconstriction, the parasympathetic nervous system causing vasodilation, at least in most vessels, right? Works that way. And it's due to that smooth muscle layer in the tunica media. And so that tends to be the predominant layer that we're seeing in arteries. This has so many implications. Things like I mentioned, blood pressure, afterload on the heart. Remember, like the resistance that the heart has to push or work against. Things like where blood flows through capillary beds, even things that we talked about a couple chapters ago, like hemostasis, blood clotting. We're going to use vasoconstriction to try to stop the bleeding until a platelet plug can form. Anyway, so vasoconstriction, vasodilation is going to be super important in here. Okay, elastic arteries. The name tells you that these have more elastic fibers, right? So we see things emphasized, like even in the tunica intima, we're seeing that internal elastic layer. This tunica externa is going to have more elastic fibers than we might find in some of our other vessels. These elastic arteries are sometimes called conducting arteries. They're going to be big tubes, like an inch, things like your aorta. are going to be elastic arteries, or your pulmonary trunk, pulmonary arteries. The nice thing about having more elastic fibers in here is going to be that these vessels are able to tolerate changes in pressure. So every time the heart contracts and it sends this bolus of blood out, right, the aorta has to stretch to accept that and then kind of recoil. If you didn't have all these elastic fibers, one, it would be super hard on the vessel, and two, you would see these big like peaks in blood pressure and falls, peaks and falls. And these elastic fibers are going to start to help kind of dampen the differences between what we see at systole and what we see at diastole. If we move down to these muscular arteries, the name is going to tell you, right, we're dominated here by tunica media. Muscular arteries are sometimes called distribution arteries, right? They're the ones that are actually sending blood out to muscles, to the viscera. A lot of the vessels that you dissect in lab are going to be these distributing arteries. If you're familiar things with pressure points, so like brachial artery, femoral artery, all of those happen to occur in muscular arteries. I shouldn't say happen to occur. They make nice pressure points really for two main reasons. They're big enough that you could cut off the blood supply to a large area by holding them. And two, since they're muscular, you can really put pressure, you have to put pressure on them to collapse them. You probably couldn't produce enough pressure to actually close off an elastic artery, and an arterial doesn't deliver to a large enough area to make a difference. I digress. Muscular arteries. Tunica media. Okay. The arterial, then this is going to be a very small artery. These are sometimes referred to as resistance vessels. This is where probably the most important vasoconstriction and vasodilation take place. Notice what we're missing here. They're not even showing a tunica externa. If there's any connective tissue on the outside here, it's poorly defined. It's not in this nice layer or tunic. The other thing I would point out here, the tunica media isn't really much to look at either, really just a couple cell layers thick. But because this is such a small vessel, we can make a big difference in things like blood pressure. Again, they're going to change diameter in response to things like local conditions, maybe listening to the sympathetic or parasympathetic nervous system. And so we'll go into that more towards the end of the chapter. Same pictures. This is from your book, Elastic Arteries. They're pointing out here that the tunica media in an elastic artery has a lot of elastic fibers, right? More than even smooth muscle. Here in the muscular arteries, we're looking again at the tunica media being predominant. And then they skipped to veins. So we're going to head over to the venous side of things. Same three layers: tunica adventitia, tunica media, tunica intima. Okay. It would make more sense to talk about capillaries first, wouldn't it? Right out of the arterioles, blood's going to the capillaries. We'll do that, and we'll come back. So the capillaries, again, diffusion is key at the capillaries, right? Taking place here. It's essentially just an endothelial tube, right? Simple squamous epithelial cells making a tube, endothelium, surrounded by a basement membrane. So not a super complex structure. But what's important is what's able to get in and out of these capillaries. Because if you think about it, so here's our options, right? If you have a continuous capillary, what that means is that the adjacent endothelial cells have tight junctions between them. It means you have a tube that is sealed and things shouldn't really be leaking out. Now, that doesn't mean that things can't leave a continuous capillary, but in order to do that, they need to actually move through the cell, which means they have to get through two cell membranes, two phospholipid bilayers. So things that are lipid soluble are gases like oxygen and carbon dioxide, not a big deal. They can actually just kind of melt right through cell membranes. And so oxygen and carbon dioxide move freely across continuous capillaries. So do things like water and small water-soluble molecules. That phospholipid bilayer just has channels, right? And so we can move in and out. But anything bigger than that, its movement really is going to be restricted. So we have a couple other options. We have fenestrated capillaries, and then I'm totally in the way here. We have sinusoidal capillaries. So fenestrated capillaries, notice that they basically have pores. They have pores that let larger molecules through. So you would find these in places like the small intestine, where all those nutrients are getting absorbed, the choroid plexus in the brain, where cerebral spinal fluid is forming, filtration sites like the kidney. Okay, so super common. Sinusoidal capillaries, you'll notice, have these big gaps between adjacent cells, and it's basically, the basement membrane is basically lacking. So notice here, even when you have those pores, there's still a basement membrane that substances need to move through. Sinusoidal capillaries are going to be super leaky. So places like the liver, the spleen, the bone marrow, some different endocrine organs is where you might find those sinusoidal capillaries. Okay, again, I do like these pictures, right? A continuous capillary, no gaps between the cells. Fenestrated capillaries, the smoke. Fenestrated capillaries have pores in them, and then the sinusoid, well, where's sinusoidal? There we go. Sinusoidal capillaries. Fenestra is just another way to say like a gap or a hole, and so it's much larger in these sinusoidal capillaries. Okay, same pictures. Here we go. Now, in capillaries, right, we said these are actually the predominant vessel. You have more of these than anything else. They don't, it's not just like you go from an arteriole to a capillary to a venule. You enter into what's called a capillary bed. And so what we have here is a single arteriole that actually is leading into dozens of different capillaries, right? So there's some blood could come in here or down here, right? So you have this capillary bed. So there's a couple things going on here, right? You actually only have enough blood to kind of fill something like 25% of your capillaries at any one time. And so what we're going to do is at the opening to these capillaries, you have sphincters. So, and notice here what they're showing is it's what's called a thoroughfare channel. So blood can just kind of bypass the capillary bed, or it can kind of keep diving off into different sides. But you have these precapillary sphincters. You have muscle cells, essentially, that can close off portions of the capillary bed and open up other portions of the capillary bed. And so you can actually do something that's called vasomotion, where you perfuse a part of the capillary bed and the next part, and then it's kind of like doing the wave, right? So this guy opens while everybody else is closed, so that gets perfused, and then that closes down, this one opens, etc. And it turns out, like when you start exercising and things, that ability to kind of open parts of the capillary bed, the coordination that's necessary to make that happen actually improves as you need it to. Okay. Oh, other thing I wanted to say, so exercise again, right? Say you've taken up running or some, very cardiovascular activity. What you'll find is that as those muscle cells are working more and more and they need more and more oxygen, they can actually release, there's a growth factor, Harrican communication, right, called vascular endothelial growth factor, or VEGF. And this actually stimulates angiogenesis, the formation of new blood vessels. And so you can actually put more capillaries into an area that's requiring more oxygen. Okay. Picture from your book, same thing. They're showing those precapillary sphincters. That's actually kind of nice, right? And so again, you can close those down, open them up to move around. Okay, and again, this picture, is kind of showing what's going on then at the capillary bed. So let's talk a little bit about diffusion. We're spending a little more time here in capillaries because this is where the good stuff happens, and then we'll hit venules on the way out. So when we talk about this diffusion taking place at the capillaries, realize we're also dealing overall with what we would call filtration. So filtration is a different process than diffusion. Filtration is what you do when you make coffee, right? You have solids on one side of a filter, and maybe then those solids are interspersed with liquid. And we see the liquid and maybe some of the smaller particles, some flavors, things like that coming through and leaving the big stuff behind. That's filtration, like letting some stuff through and leaving the solutes behind, or the bigger things behind. So what we're going to see happen, right, is things that like red blood cells, white blood cells, and then proteins are pretty big. Those sorts of things are all going to stay in the blood, but some of the smaller solutes, water soluble things, maybe sodium and chloride, are able to leave the vessels at the capillary bed. Okay. This is the picture that your book gives, and then I'll show you why I kept the other picture, right? So when you're looking at what's going on in a capillary, they're going to talk about net filtration pressure. And again, if we're leaving the solids, things like our coffee grounds or blood cells inside of the vessel, we're talking about how much coffee we make. But what we have to realize is that we've got filtration taking place, but also reabsorption. Okay. And so what you're going to have to look at is what's causing the filtering to take place and what's causing the reabsorption to take place. And I think the picture from the previous text is going to do a little bit better job at this. Okay. So we've got some terminology here. So when you see hydrostatic pressure, this is a pressure that a fluid exerts. Right? Hydrostatic pressure. This is the pressure of a fluid. For this particular example, right, they're talking about capillary hydrostatic pressure or blood pressure, right? How much pressure, how much force is there that's trying to push fluid out of this capillary, right? What's encouraging that filtration? It's not as simple as just saying, well, what's the blood pressure? We know then how much filtration, because the other big force that's working here is what's called blood colloid osmotic pressure. Now, anytime you see a colloid pressure, or sometimes you'll see that called osmotic pressure or oncotic pressure, right? We're talking about the attraction of water towards a molecule, right? Whether that's a salt or something larger like a protein, there's an attraction and the water is getting pulled back towards that. So while we have blood pressure trying to force things like maybe some of the plasma out of the blood supply, what we're leaving behind, all those blood cells and proteins, are actually acting kind of like a sponge. They're pulling those fluids back. And so we see that if the blood pressure, the hydrostatic pressure, is bigger than that colloid osmotic pressure, that we get filtration taking place. And this happens all the time, right? There should be fluid leaving the capillaries and kind of bathing the tissues. It speeds up the movement of things like hormones and nutrients, all this. But reabsorption is super strong too. So notice they're actually pointing out that every day you filter about 24 liters of fluid out of your blood, but you reabsorb 20 of them, right? So it's really a net 4. But it goes out and gets pulled back in. It is going to get pulled back in when that colloid osmotic pressure exceeds the hydrostatic pressure. And that tends to happen down on this venous end because all of the fluids that you've pushed out, you've left but the sludge behind. And so by over here, right, it's all just like proteins and red blood cells. And so there's a really strong blood colloid osmotic pressure on this end, right? And so we see if blood colloid osmotic pressure is greater than capillary hydrostatic pressure, we see that movement back into the capillaries. Okay. It's slightly more complicated than this because There's also a tissue hydrostatic pressure, right? So the tissues, as a lot of water starts to build up in that extracellular fluid, it would actually exert a force that forces it back into the blood. And then there's also a tissue osmotic pressure where there's salts and proteins out there. So it's a little more complicated than this, and you'll see that as you move into some things like pathophysiology, but this is big picture. If you know what a hydrostatic pressure is and a colloid or colloid osmotic pressure is, that's going to help you hugely. The other thing to note here is this excess, right? So we said we filter 24 liters a day and we've reabsorbed 20 of that. Well, what happened to the other 4? It actually gets picked up by the lymphatic vessels, and we're going to talk about those next week. But this is going to be a whole system that's set up to kind of recollect these fluids and return them to the circulation. But the nice thing is we're going to put them through a filter first. We're going to put them through lymph nodes and those certain other lymphatic tissues, places like the spleen. Okay, so again, when this happens across the capillary, some important things, right? We're actually exchanging plasma and extracellular fluid. This accelerates the distribution of nutrients, hormones, things like that. Proteins that are too big to just like diffuse out of blood vessels are going to stay in the circulation, right? This can also help us flush things like toxins, pathogens, and wastes out to the lymphatic system. So there's a lot going on. We're not just making coffee. Okay. Right? I mean, way more than this picture is showing you. Okay, so that's our capillaries. Let's keep going. We got to return this blood to the heart. So after the diffusion is taking place at the capillaries, regardless of what kind of capillary, we're going to move into a venule. Venules are very similar to capillaries, right? So you might notice it's basically a capillary with now a thin connective tissue, tunica externa. It's completely missing that tunica media. Occasionally you'll find like a smooth muscle cell here or there, but there's really not much going on. In a medium-sized vein, you have a very thin tunica media, okay? So again, the smooth muscle layer. The tunica externa is much more dominant here. So if you think of these vessels as being like the same size, notice that artery. It's the tunica media that dominates. In the venous side, it's going to be the tunica externa on a vessel of the same size. And this really comes down to the venous side of this is under way less pressure, okay? We'll still see, you can do, it's called venoconstriction and venodilation. You can still do this on this side, but it doesn't come into play near as often. From there, blood's moving into these larger veins. Again, thin tunica media, thick tunica externa, which has lots of collagen, elastic fibers, etc. The cool thing, so then I'm like, oh, they're kind of boring, the veins, but they got a couple cool things. They do have valves, right? So again, this side of the circulatory system is under such low pressure that the blood pressure can't actually overcome gravity. And so blood wants to keep like pulling down towards your feet. And so you're actually using the skeletal muscle in the legs here to propel the venous blood back towards your heart. And then between that muscular activity, the blood wants to kind of settle back down, and these valves are closing and keeping the blood moving in one direction. When you get back up to the kind of like the thoracic cavity, think about, this is crazy, that inferior vena cava, huge vein, huge amount of blood trying to return to the heart. And then instead of it being, say, like the calf muscle, they actually talk about the respiratory pump. Each breath you take is actually working to return that blood to the heart, which I think is kind of cool. Okay, so what it ends up looking like, if you looked at the distribution of blood and where it's sitting at any time in the cardiovascular system, this is what we would see. There's actually a lot more of it in veins than there are in arteries. The veins have a larger compliance. They're more open, and they can kind of act as a reservoir, kind of act as a pool. And so that's one of those places where, like, if you suddenly had a lot of blood loss, you could actually have venoconstriction, and the veins would constrict and try to improve your blood pressure, move more of that blood back to the arterial side of things. Okay, I think let's take a break there, and then we'll come back and we'll do some physiology.